Hello students, welcome back. Today we'll see the basic concepts about gene. So now you all know that chromosome is made up of nucleic acid and proteins, and DNA is a genetic material. So now actually within DNA linear set of genes are present, and that genes are hereditary material. So first of all we see some definitions of genes. A specific DNA sequence that contains genomic information is called gene. So of course, gene is nothing. It is a small part of DNA sequence, and that particular part of DNA sequence, which contains genetic information, which codes for amino acids, which have codons in it, that particular part is called gene. Gene is unit of genetic information that specifies the synthesis of polypeptide. Now, within DNA, at specific locus, genes are present, and that gene have information in it, information of proteins. So that gene, it codes for particular amino acid, and amino acids combine to form polypeptide. The unit of genetic information that controls a specific aspects of phenotype. So gene, it has an information. It has an information of what? It has an information which codes for protein, and finally it produces phenotype. Thus, as it controls phenotype, as it has a hereditary material, it has a genetic information, it is called unit of heredity. Now we'll see history. So first of all, gene was coined by Johnson in 1909. Garrett proposed one gene, one product hypothesis in 1908. That is, one gene can produce one product. One product means one protein. That was proposed by Garrett in 1908. Gene theory was proposed by T.H. Morgan in 1911. In his theory, he suggested that chromosome carries heredity unit, and each chromosome carries hundred to thousands of genes in it. And the genes are arranged in a linear manner in chromosome and at a specific site which is called locus. So this was the concept given by T.H. Morgan. He said that within chromosome it carries genes which are linearly arranged at specific site which is called locus, and within one chromosome there are hundreds to thousands of genes present. So we can say that number of chromosomes far exceeds number of genes far exceeds the number of chromosomes. Pauling and Ingram in 1949 established the role of genes in protein synthesis. So now we know that gene have information within it, that is codon codes for amino acid, and that produces protein. But Pauling and Ingram in 1949 at that time established that there is a role or there is link between genes and protein synthesis. Beetle and Tatum in 1948 proposed one gene, one enzyme hypothesis. That is, one gene can produce one enzyme. That hypothesis was given by Beetle and Tatum in 1948. The fine structure of gene was proposed by Benzer in 1962. Now, structure of gene. So gene consists of mainly three parts: coding region, which is called exons, non-coding region, which is called intron, and a regulatory sequence. Coding sequence, which is called exon, it specifies a sequence for amino acids, while non-coding do not specify amino acid, and regulatory sequences have a role in regulation of process. So as you can see in diagram, this portion, dark one, that are exons, exon 1, 2. Between two exon, intron is given. So this is intron, this is exon. So this is non-coding region, this are coding region, and upstream to this genes, this are genes. This is a complete gene in which coding region, non-coding region is present, and upstream to that gene are regulatory sequences which is involved in regulation. Now we'll see certain features of gene. First of all, location of gene. Now we know that genes are present within DNA, that is, inside cell and nucleus within chromosome. Genes are linearly arranged. Genes, they are transmitted from parents to offsprings. That is why characters, or you can say phenotype, is developed. They are responsible for character of organism. The site at which genes are present on chromosome is called locus. Several genes are located on each chromosome. So, as I told earlier, as in a single chromosome, many genes are located. The number of genes far exceeds the number of chromosomes. The genes are linearly arranged. Now, genes can undergo sudden heritable changes, that is called mutation. Mutation means change in your genomic material. So, gene can cause that. So, mutation can be undergone in this genes. Due to mutation, if sequence changes, that is position of gene or sequence in a gene changes, then character of genes also changes. Genes exist in several alternative forms, that is called allele. We already have discussed the term allele. Allele means alternative form of same gene. That is, in a diploid organism, two sets of chromosome are present at the same locus. Same gene is present which codes for same character, but it can produce different effect. Such alternative form of gene is called allele. Gene can undergo duplication, that is, it can produce its ditto copy, and that process is called replication. It can even undergo process of recombination, that is, two different sequences can recombine to produce a new one. The process is called recombination, and genes are capable to undergo recombination. A gene is a particular DNA segment which contain information to synthesize polypeptide chain. This we have earlier discussed, but now let me explain you with diagram. So this is a complete cell. Within that, this is a nucleus. This actually is a DNA, and within DNA, this portion given is gene. Within gene, exon, that is coding region, and introns are present. Now, that is coding and non-coding region present. Through transcription, RNA is produced. After transcription, post-transcription modification occurs, and in one of the process of post-transcription modification is RNA splicing. That is, unwanted region of RNA, non-coding sequence of RNA is spliced. A complete mRNA is then produced. So you can see over here, after RNA splicing, mRNA is produced within nucleus. That is, replication and transcription, two important processes of central dogma of life, occurs in nucleus in eukaryotes. And the last process, that is protein synthesis, it occurs in cytoplasm. So mRNA, once it is synthesized, it is exported from nucleus to cytoplasm, and it reaches to the site where protein synthesis occurs, that is at ribosome. So here you can see protein synthesis occurs from mRNA. So actually, gene has an information to code protein. The information which is present in gene is contained as a sequence of nucleotide, which is called genetic code. So as we know, gene is nothing, it is a part of DNA only, and DNA is made up of nucleotides. So the information within gene present is in a form of nucleotide, and that is called genetic code. The sequence, or you can say genetic code, they are in the form of three nucleotides that codes for a single amino acid, that is triplet triplet. Three nucleotides, a triplet, a single triplet combined to form one codon, and one codon codes for one amino acid. See, you can see over here in a table, these are different codons given in different combination. That codon, it codes for particular amino acid which is also given. You can see over here leucine, methionine, lysine, glutamic acid. So this has the codons which produces different amino acids. So another definition of gene is regarded as fundamental, indivisible unit of information, genetic information in terms of structure and function both. So at that time, it was known that the gene is the smallest unit. It cannot be further divided, so indivisible term was used. Further, this was challenged by recombination studies in bacteriophage by Benzer, and they showed that it could be subdivided into smaller units. Gene can further, again, be divided into smaller subunits, which are muton, cistron, and recon. Muton is the smallest portion of gene which can undergo mutation. Recon is the smallest portion of gene which can undergo recombination. You can say function of recombination and muton as function of mutation. While cistron is the smallest part of gene which codes for amino acid. And in gene, non-coding sequences are also present. So the portion which only codes, they are called cistron. So what is cistron? Again, that is an important terminology. It is unit of function. Why unit of function? Because its function is to code amino acid. So that portion of gene which can code, which have function to produce protein, that part is called cistron, which can synthesize one polypeptide. So that part which can synthesize peptide. This, thus, cistron is called the smallest unit that transmits genetic information. So finally, the smallest unit which can transmit genetic information is termed as cistron. But in case of prokaryotes, cistron and gene both are similar. But in case of eukaryotes, as eukaryotes contains introns, gene and cistron two terminologies differs. So in prokaryote, cistron is an alternative term of gene. In prokaryotes and in lower eukaryotes, there is usually a simple relationship between gene and its product. That is, as introns are not present, between within gene, inside one gene, non-coding sequences are not present. Directly that gene directly codes, it produces mRNA and it directly codes for protein. So there is a simple direct equivalence relationship between gene and ancestron. Thus, gene and cistron are equivalent. That is, you can say gene is a unit of genetic information in case of prokaryote, or cistron is a unit of smallest unit of genetic function. See, here in diagram it is given. This is single gene, that is gene 1, gene 2, and gene 3. In between two genes, internal UTRs, that is untranslated regions, is present. But within gene, non-coding sequences are not present. Between two genes UTRs are present, but within gene, non-coding sequences are not present. So this DNA is transcribed to produce RNA, and that RNA further is translated to produce amino acid. So directly one gene produces one protein, second gene produces second protein. So gene is directly proportional to its product produced. That is why this gene is called cistron. But in case of most higher eukaryotic gene, it contains intron. In eukaryotes, within coding sequences, or you can say within gene, introns are present. That is, genes are interrupted by non-coding sequence. A complete stretch of coding sequence are not present. Between coding sequences, non-coding regions are present. Genes are interrupted by non-coding sequences, which are not presented in final product. Now, this non-coding sequences, that is introns, they are spliced out. They are removed by post-transcription modification process, and that non-coding sequences, as they do not code for amino acid, they are not present in the final product, that is in protein. Therefore, they are not part of its function. Only coding sequences, exons, are the part of its function, that is, they are the part of protein, and introns are not. Thus, cistron is therefore equivalent to exon of eukaryotic gene. So, in case of prokaryote, cistron and gene are equivalent. But in case of eukaryote, cistron and gene are not equivalent. Cistron is equivalent to exons